Welcome to this tutorial to demonstrate basic functionalities within the R2 Genomics Analysis and Visualization Platform. In this tutorial, we will take a look at the View a Gene page. The View a Gene function is one of the core modules to do in-depth analyses within one or two genes, also named reporters, within the platform. From the main page, you can select View a Gene as the analysis option in box 3. Click Next to progress and provide settings to inspect your gene of interest. We can now specify the gene that we would like to inspect. We start by typing the gene name in the Search by Gene field. In this example, we will look at the mikin gene, an oncogene transcription factor that is well known within the neuroblastoma community. Once we start typing the gene, a drop-down will appear with the genes that match the description. We click on one of the suggested reporters to select it. There are many options that can be adapted from this representation as well, but we will get to this later. We press Submit to view the gene. We have now reached the one-gen view for our gene of interest. In the center, an image is shown where all the patients from within this cohort are represented. The samples within this data set are ordered according to the expression of the mikin gene, from low expression at the left towards high expression to the right. If we hover over the blue-colored dots, then annotation provided for these samples will be displayed in pop-ups. Below the samples, we show a number of annotations, also named tracks, within R2 in color-coded boxes. These tracks allow us to quickly see associations between the expression of our gene of interest. Below the image, a form is shown where many things can be adapted. For example, we can change the tracks that we want to show at the bottom of the picture. To adapt the tracks, we simply click on the Select Tracks button. A tree will load, where we can toggle which tracks to represent. We deselect all tracks except the mikin amplification status and press Submit. The image now becomes less busy, only showing the amplification status. In the form, we can also simply change the gene that we could look at. In the field of the gene, we now type ALK and press Submit once more. We now look at the cohort from the perspective of ALK. We can also combine these two genes in a plot that we term Y-plot. From the Analysis Type drop-down, we select Gene versus Gene. A second gene selection item becomes available. Here we type mikin and select the reporter. We press Submit again and a new image is generated. The samples are now sorted on the expression of ALK, and in another color, the value for mikin is added to the view. If we click on Swap Genes, then the perspective will change to mikin again. We can again also change the genes we want to look at once more. We will look at the R2 gene and press Confirm again. Below the image, we can also see the correlation between the two genes. Here, the association is not very strong, and in fact is also not significant, as can be seen from the p-value. It may be a bit difficult to assess the correlation from the Y-plot perspective, especially if you're getting started with R2. We can also change the type of graph from the form settings. Let's change the plot to an Z-plot. Here we can also clearly see there is no correlation. In the form, we can also color the samples by a track. Let's activate the mikin amplification status. In addition, we click on the More settings and switch Linear Fit to On. We press the Confirm again. Now this starts to look interesting. Because we use the Linear Fit, we can now also see that the relation between the two genes is very significant in the non-amplified group, but even shows a negative correlation in the amplified group. We hope this short demonstration has been helpful to get an idea how the R2 Genomics Analysis and Visualization Platform works and how easy it is to perform consecutive analyses to identify something interesting.